Here's the third part of Welcome to part 3 of chapter 8. Mutations. We know that mutations are changes in DNA sequence. Has to start with the DNA. Changes in DNA sequence. How might this happen? Changes in DNA sequence can be replication errors. And what's replication? That's when we copy DNA. S phase of interphase. Right? It's supposed to be idiot-proof. It's supposed to make no mistakes. If there's an A on the template, DNA polymerase is supposed to add a T. If there's a T, we add an A. If there's a G, we add a C. And if there's a C, DNA polymerase adds a G. Sometimes mistakes are made, and a C might be placed with an A by mistake. That is a mutation. There are also mutations that are damage to DNA. So UV light, UV damage from the sun damages DNA. X-rays can damage DNA, and other chemical mutagens and carcinogens, things that are associated with cancers. Cancers are caused by mutations in DNA. Usually lots, not just one. We estimate that perhaps 1 in 10 million base pairs contains an error or a mutation, right? That would be an error. That's not fixed by the ability of these enzymes to proofread. So DNA polymerase can fix some of its own mistakes, but some mistakes also slip through. Some of the changes in DNA sequence, otherwise known as mutations. What types of mutations are out there? Again, the definition, unrepaired change in the sequence of your DNA. A point mutation just means a single base pair, a single, what is that also called? A nucleotide. One of the G's, the C's, the T's, or the A's is wrong. If it's a substitution, it means one base is replaced by another one. That's my example of it's supposed to be a T, but by mistake, polymerase adds an A or a C or a G. An insertion is when there's an extra nucleotide added. So there's one too many. A deletion is when one is removed, one too few. So plus or minus. Here are some examples of insertions and deletions. Could be point mutations. Remember, that's one single base, or involve many bases. And here's our example. If we have the normal sequence, if this is the DNA, and we make an RNA, which makes a different protein, right? These are the amino acids in your proteins. If we have a change in DNA sequence, so this right here, this T, right, was supposed to be a C. That means this would be an A here in the RNA because of the complementary base pairing. That would change which amino acid is added to the protein. That would change what that protein can do, or even if it's functional. So that's a substitution, a T instead of a C. An insertion is when we actually get a complete extra one, right? The original sequence CTT is still here, CTT, CT, CTT, CT, but we've added an extra C. That changes all the amino acids going down into your protein. If we delete one, we take one out, we remove an A right there, then that again shifts the reading frame to make our protein and changes our protein's amino acids. Another type of mutation that can happen is called a silent mutation. That's usually when there is a change in the DNA, so that A is now a G, that changes the sequence of the mRNA. There's a U, this became a C, but it doesn't change what amino acid goes in the protein. So even though it's a mutation at the DNA and RNA level, it's not at the protein level. So it's a silent mutation. You'd never know if you looked at the protein. Sickle cell disease is an example of a substitution mutation. In one single nucleotide. In normal hemoglobin, that's the protein that is in our red blood cells that helps us carry oxygen. This T is substituted with an A. That causes the shape of the protein to change, which causes the overall shape of the red blood cell to change. They call it a sickle because that looks like the shape of something we call a sickle that used to be used to cut grain, in that shape of a knife. And it's pointy. See how pointy these are? Right? These red blood cells do not flow through your blood vessels very well, especially small capillaries. They get stuck, causes lots of pain. If your red blood cells are stuck and they're not moving around your bloodstream, delivering oxygen, you have low oxygen in your blood that can cause you to faint and even die. So if we were looking at this example, what would we call this? What type of point mutation is it? If this was supposed to be a T here and an A, and it was mutated to this T mutated to an A, what would we call that? Substitution. One nuke for another. Sickle cell disease is an autosomal recessive disorder. Remember what that means? You have to have two alleles, homozygous recessive, in order to have the disease. So how many mutated hemoglobin alleles do people with sickle cell disease have to have? Two, both of them homozygous. We alluded to repairing the DNA mutations at the beginning. DNA polymerase can repair its own mistakes, so it proofreads or it edits itself. And if it sees that it added a T where it was supposed to add a C, then it can remove it and correct it as it's doing DNA replication. So during DNA replication. If any of these mistakes get through replication, the proofreading mechanism of polymerase doesn't work, then there's other repair proteins that fix almost all of them. 99% are fixed when they're made mistakes. But that 1% that gets through can still cause new alleles, can cause disease, or it can be totally neutral. So one remaining mistake for every billion bases. That's really, really good. But that doesn't mean they don't happen because we have trillions of base pairs in our chromosome. So the question is DNA repair 100% effective? No, just told you it's not. Silly. 99%. What would happen if DNA repair was even less effective? More mutations. Maybe more disease, maybe more death, maybe more new alleles that are good. It's hard to tell. This is just a picture here of how some mistakes are removed. You do not need to know the mechanism of this, but please know that we do repair most of them, but some still slip through. If we were 100% effective at DNA repair, cancer would not exist. It does, which means we're not. And there you go. There's mutations, the end of chapter 8. Thanks, guys.